Welcome to the deep dive. Today, we're digging into something pretty critical in cardiac fibrosis.
hmm
The source material we're looking at explores a really fascinating preclinical strategy using engineered T cells.
Yeah, it's a big one. Fibrosis is, well, it's the common denominator in so much heart disease, driving heart failure.
Mm-hmm.
And honestly, our current toolkit to directly tackle it is pretty limited.
Exactly. So, finding new angles is crucial. Our mission here is to really unpack this research, how they're trying to use these engineered T cells to specifically target and hopefully resolve that damaging fibrosis.
And they came at it from two angles, which is smart.
Right.
First, a kind of proof of concept using a foreign antigen just to show it can be done.
Okay.
And second, identifying and targeting something native, an endogenous protein called FAP, fibroblast activation protein. That's the more clinically relevant piece.
All right, let's start with that proof of concept then. The foreign antigen was ovalbumin, OVA. How did they get that only onto the activated fibroblasts? That seems tricky.
It is, yeah. They used a clever genetic tool in mice, basically a tamoxifen-inducible Cre recombinase,
Okay.
driven by the periostin promoter. Now, periostin ramps up specifically in activated fibroblasts during cardiac injury.
So the periostin acts like a flag on those specific cells.
Exactly. And the tamoxifen is the trigger. You give tamoxifen, the Cre enzyme gets active only in those periostin-expressing fibroblasts, and that switches on the expression of the OVA peptide.
So you get OVA displayed just on the cells you want to target, the ones involved in the fibrosis.
Precisely. A molecular tag appearing only after injury and tamoxifen treatment.
And the injury itself, how do they induce the fibrosis for this model?
They used a pretty standard approach, continuous infusion of angiotensin II and phenylephrine, Ang-IP.
Right, the Ang-IP model, known to cause hypertrophy and fibrosis.
Yep, for 28 days. They checked and confirmed significant fibrosis was already established after just 1 week of this infusion. That's important. They weren't preventing fibrosis. They were treating existing fibrosis.
Okay, so fibrosis is there. Then come the T cells, these OTI cells. What's their deal?
They're CD8 plus T cells, killer T cells, taken out and specifically engineered to express a T cell receptor that recognizes that OVA peptide.
So they're like heat-seeking missiles for any cell showing OVA.
That's a good way to put it.
Yeah.
They transferred these OTI cells into the mice 1 week after starting the anti-IP, right when that fibrosis was kicking in.
And crucially, controls. You need to know it's the T cells doing the work.
Oh, absolutely. They had mice with the anti-IP and tamoxifen, so they expressed OVA but got no OTI cells.
Okay.
They also had mice that could express OVA generally, but didn't have that periostin-driven Cre, so OVA wouldn't be specifically on the fibroblasts, even if they got the T cells. Central controls.
Makes sense. So the million-dollar question for this part, did it work? Did targeting OVA on fibroblasts reduce fibrosis?
It did, quite convincingly. At the 4-week mark, the mice that got the OTI cells showed a really significant reduction in cardiac fibrosis compared to all the control groups.
Wow. Okay.
And interestingly, they also saw a partial rescue of the cardiac hypertrophy. The heart weight to body weight ratio improved somewhat.
So proof of concept achieved. You can use the immune system to target and clear activated cardiac fibroblasts.
Exactly. It demonstrates the principle is sound. But, targeting OVA isn't a therapy you can take to humans.
Right. Not practical. So that leads perfectly into the next phase, finding a human target, something endogenous.
Correct. They needed a marker present on activated human cardiac fibroblasts in disease, but ideally absent or very low on healthy heart cells and other organs. That brought them to FAP.
Fibroblast activation protein. How did they land on FAP specifically?
They leveraged human data, looked at RNA sequencing from actual human heart tissue,
From
non-failing donor hearts compared to hearts from patients needing transplants due to hypertrophic cardiomyopathy, HCM, or dilated cardiomyopathy, DCM.
Okay, diseased versus healthy.
And they were hunting for genes turned up high, specifically in fibroblasts within those diseased hearts.
Looking for that pathological fibroblast signature.
Exactly. And FAP just jumped out. It showed the biggest fold change increase in expression in both DCM and HCM compared to the non-failing controls.
A strong signal then. What do we know about FAP biologically? Why would it be a good target?
Well, it's a protein found on the cell surface, which is good for, say, CAR-T targeting. It's a protease involved in remodeling the extracellular matrix.
Okay.
Its expression is known to go up during development, wound healing, fibrosis, and also in tumor stroma. But, and this is key, it's generally very low in most normal adult tissues.
Ah, that restricted expression profile, that's the therapeutic window you need.
Precisely. Minimal expression under normal conditions is critical to hopefully avoid attacking healthy tissues.
Did they confirm this pattern at the protein level in the human heart samples too, not just RNA?
They did. Immunohistochemistry confirmed strong FAP protein on cardiac fibroblasts, importantly, not on cardiomyocytes in those failing human hearts, and minimal FAP in the normal hearts.
Okay, RNA and protein data aligning nicely. FAP looks like a solid candidate.
Very promising endogenous target based on that human data.
So, next step. Back to the mouse model, but this time targeting mouse FAP instead of OVA.
Exactly. They went back to the NGIP model.
Yeah.
First, they confirmed that, yes, just like in humans, FAP expression was strongly upregulated on activated cardiac fibroblasts in these mice after a week of NGIP.
And importantly, low elsewhere.
Yep, minimal FAP in healthy mouse hearts and other organs. They also checked other injury models, MI, TAC, and saw FAP upregulated there too, suggests it's a fairly common marker of fibroblast activation and injury.
Good validation. So time for the FAP CAR T cells.
Time for the FAP CAR T cells. They engineered CAR T cells to recognize mouse FAP.
Okay.
And they gave these to the NGIP mice adoptively at week 1 and week 2 after starting the NGIP.
2 doses. Why 2?
Likely based on experience from tumor models where mouse CAR T cells sometimes don't persist that long in vivo. So second dose to ensure sustained pressure.
Makes sense. Did they confirm the CAR Ts actually got into the heart and found the FAP positive cells?
They did. They show the FAP CAR T cells infiltrated the myocardium and, crucially, were co-localized right there with the FAP-expressing fibroblasts. Direct evidence of target engagement.
Okay, the moment of truth again. What happened to the fibrosis and heart function in the FAP CAR T treated mice?
The results were really encouraging. At 4 weeks, significant reduction in cardiac fibrosis compared to controls.
Similar to the OVA result.
Yes, a strong antifibrotic effect.
Okay.
And importantly, they also saw a partial rescue of cardiac function, both systolic and diastolic parameters, improved on echo.
So hitting FAP didn't just reduce scarring, it actually helped the heart work better. That's huge.
It's a key finding. Structural improvement linked to functional benefit.
But I recall the paper mentioning some fibrosis remained, specifically around blood vessels, perivascular fibrosis.
That's a very astute point, yes. They noted that while the interstitial fibrosis, the scarring between muscle cells, was reduced, the fibrosis around the vessels seemed less affected.
Why would that be?
It correlated with their finding that those particular perivascular fibroblasts in this NGIP model didn't express much FAP.
Ah, so the CAR Ts couldn't see them.
Exactly. It highlights the specificity of the approach. It only hits FAP-positive cells. It also points to potentially heterogeneity within the fibroblast population. Not all activated fibroblasts might express FAP, at least in this model.
Interesting nuance. So FAP is effective, but maybe not the whole story for all types of fibrosis. Now the big one, safety. CAR T therapy can have significant side effects. What did they find?
They did a pretty thorough safety workup. Histology on numerous non-cardiac organs showed no signs of damage, consistent with that low baseline FAP expression elsewhere.
Okay, good start. What about systemic inflammation, cytokine release?
They tracked serum cytokines for 12 weeks. NGIP itself raises cytokines, as expected, but adding the FAP RT cells didn't significantly push those levels higher over the long term, suggests it wasn't triggering a massive systemic storm.
That's reassuring. Any signs of trouble within the heart itself? Cardiotoxicity?
They looked at gene expression in the heart using a cardiotoxicity array. There were some changes at 4 weeks, but the pattern wasn't clearly toxic, and importantly, those changes mostly normalized by 8 weeks.
So maybe some transient inflammation, but resolving.
Seems like it. They also looked at a broad panel of immune and inflammation genes. Only about 22 genes showed changes at 4 weeks, again suggesting a relatively mild, contained response within the heart.
What about immune cell infiltrates in the heart? Did the CAR-Ts bring in a lot of other inflammatory cells?
Not really. Major changes in T cells, CD3, CD4, CD8, or neutrophils, MPO. They did see fewer B cells, interestingly, and more macrophages, F480 positive cells. The significance of that needs more study, but no red flags indicating massive, uncontrolled inflammation.
And the animals themselves, how did they fare long-term? Any other potential issues like wound healing given FAP's role there?
All the treated mice survived the full 12 weeks with no obvious signs of illness. They specifically tested wound healing and saw no delays or problems. And even though there are some rare FAP-expressing cells in the pancreas, serum amylase levels were normal, suggesting no pancreatic damage.
Okay, so overall, in this preclinical setting, the safety profile looked manageable, reasonably clean.
Reasonably favorable, yes.
Yeah.
Especially considering the potential potency here. But it's still mice, of
Of
course.
course. So, wrapping this up, what are the big takeaways here, the implications?
I think the biggest implication is that this provides really strong preclinical validation for using engineered T cells immunotherapy against cardiac fibrosis. It opens a potential new therapeutic door.
A whole new modality for heart disease, potentially.
Potentially, yes.
Right.
But, and it's a big but, there are many hurdles before this gets near a human patient.
Right. What are the key challenges ahead? What needs to happen next?
Well, first, is FAP truly the best target in humans? Across different types of heart disease, we need more validation, maybe combinations of targets. Safety, safety, safety. We need rigorous testing in larger animals to really probe potential off-target effects, which might be subtle in mice, but significant in humans.
The SAR construct itself probably matters a lot too.
Huge impact. Which co-stimulatory domains you use, the linker, the binder, all these can affect efficacy and persistence, but also safety. Lots of optimization needed there.
And the risk of long-term side effects or unforeseen toxicities.
Absolutely.
Right.
Which brings up the idea of control mechanisms. Maybe engineering kill switches into SIR-T cells so you can eliminate them if things go wrong. That's being actively explored in oncology and would likely be crucial here too.
It feels like we're seeing the beginnings of the immunorevolution, which has transformed cancer care, potentially starting to extend into cardiovascular medicine.
I think that's a fair assessment. It's early days for this application, but the potential is undeniable. This work really cracks the door open.
Fantastic summary. So, this deep dive has shown us compelling preclinical evidence. Engineered T cells targeting FAP can reduce cardiac fibrosis and even improve heart function in mice, a potential new weapon against heart disease.
A very exciting potential weapon, yes.
So a final thought for you, our listener, to ponder. We know cardiac fibroblasts are complex, they're not all the same, and fibrosis itself varies depending on the cause of heart disease. Thinking about moving this towards humans, what do you see as the single biggest challenge, maybe scientific, maybe ethical, in selecting the right targets and ensuring these powerful immunotherapies are truly safe and effective for treating heart failure? Something to chew on. Thanks for joining us for this deep dive.